Hello students, my name is Arif Malik and you are watching my channel Arif Biology. Today in this video tutorial we will start here a new chapter so called virus. आज हम माइक्रोबायोलॉजी का एक चैप्टर डिस्कस करेंगे वायरस. So first we will know here actually about what's actually virus. What's actually virus? Virus is actually it's an infective agent that consists of nucleic acid, that's DNA or RNA. This DNA or RNA is present inside protein coat. This is actually virus. So it's a DNA or RNA, not both. DNA or RNA within a protein coat called virus. It's a very small to see as by light microscope. We cannot see it by light microscope. It's very tough because it's very small and is also able to multiply only within the living cell of the host. Means when this virus enter into the living cell of host, then it start to multiply. Then viruses multiply. When it multiply, when viruses invade inside the living cell of the host. This is actually viruses. Or actually this virus term belongs to Latin word. Is it? This virus belongs to Latin word जिसका meaning है venom of poison. So this is a Latin word which means venom of poison fluid. Venom of poison fluid. This term was given by actually a scientist जिसका नाम था Pasteur, one of the great scientists. इसने term दी थी कौन सी? That is virus. Another point about the virus is that is a virus consists of 2 or 3 parts. Simply जैसे मैंने कहा, the first one is here, that's genes. Genes are made either from DNA or RNA. As you already know that genes is present inside DNA और RNA में भी हो सकती हैं. इसमें RNA भी होता है. Long molecules that carry genetic information. The second is part of this virus is that is protein. It's actually coat which protects the genes. That's this RNA and DNA को protect करता है. This is called protein coat. We will see it later. What's actually this protein coat? And in some viruses an envelope of fat. Some viruses में क्या होता है? Along with protein coat, there is also another layer which is called envelope. ये भी protect करती हैं किसे? That's the another point is that these viruses vary in shape. Means there is different shapes of virus. That's virus can be helical type. That's they can also be a icosahedral type. They can be complex type like bacteriophage. ठीक है ना? इनका complex structure हो सकता है icosahedral, helical. Virus range in size. इनका size भी अगर हम देखें 20 to 300 nanometers it can be. And second point about the virus is that virus spread in many ways. How virus ways? Actually, this is the spreading vectors. These are spreading vectors. How they spread? Viruses spread in many ways. Just as many viruses are very specific as to which host species or tissue they attack. जिस टिश्यू और होस्ट के टिश्यू और सेल पे अटैक करते हैं वहां पर ये क्या करते हैं? दे स्टार्ट इन्वेडिंग देयर. क्या होता है जहां पर भी ये इन्वेड करते हैं एंड दे हाईजेक दे दैट इज जीनोम ऑफ होस्ट एंड स्टार्ट सिंथेसिस इट्स ओन जेनेटिक मटेरियल. ठीक है ना? Another point is that plant viruses are often spread from plant to plant by insects, other organisms known as vectors. प्लांट्स में ये वायरस जिनके साथ होता है दैट स्प्रेड होता है दैट्स इंसेक्ट्स मोस्टली और समटाइम्स प्लांट टू प्लांट एंड जिन ऑर्गेनिज्म के थ्रू ये स्प्रेड होता है दीज आर नोन एज सिंपली वेक्टर्स. Some viruses of animals, including human virus, are spread by exposure to the infected body fluids. Sometimes body fluids can spread this virus to humans. Also, like influenza virus. Influenza virus spread through air by droplets of moisture when people cough or sneeze. During sneezing or cough, this influenza virus can spread. Is it? Viruses such as norovirus are transmitted by the fecal or oral route, which involves the contamination of hands, food, and sometimes water also. So these are vectors. That is how this virus actually spread to other organisms. The human immunodeficiency, that's HIV, is transmitted during sex. Is it? This is about HIV. Dengue virus is spread by blood sucking of insects. Rotavirus is often spread by direct contact with infected children. Antibiotics have no effect on the virus. This is important point about the virus. That's these antibiotics have no effect on the virus. But antiviral drugs have been developed to treat life-threatening infections. That's vaccines. So what's vaccine? Vaccine is antiviral drug. It's developed for the treating of this threatening organisms. And these vaccines can also boost our immunity. Now let's move towards discovery of viruses. How virus actually discover? In 1884 the फ्रेंच माइक्रोबायोलॉजिस्ट जिसका नाम था चैम्बरलैंड, इसने इन्वेंट किया था एक फिल्टर जिसे हम आज किस फिल्टर के नाम से जानते हैं, दैट्स चैम्बरलैंड पॉस्चर फिल्टर. इस फिल्टर के थ्रू दैट इसमें इतने स्मॉल पोर्स होते हैं ताकि इसमें से स्मॉलर देन मींस दीज पोर्स आर स्मॉलर देन बैक्टीरिया. दीज पोर्स आर स्मॉलर देन बैक्टीरिया. सो इस फिल्टर के थ्रू इन वायरसेस को फिल्टर किया जा सकता है. ये डेवलप किसने किया था? दैट्स दिस चैम्बरलैंड. इन द अर्ली 1890s दैट्स रशियन बायोलॉजिस्ट इवानोव्स्की यूज्ड दिस फिल्टर फर्स्ट टाइम टू स्टडी द टबैको मोज़ेक वायरस. इसने इनफैक्ट जो टबैको प्लांट था उसको फिल्टर किया इसके सॉल्यूशन को थ्रू दिस दैट्स चैम्बरलैंड पॉस्चर फिल्टर के थ्रू. And then he come with a conclusion of first virus, that's tobacco mosaic virus. In 1899 there was a great scientist whose name was Bajernik. इसने क्या किया? He discovered that these viruses can divide only in living cells. क्या इसने कहा था? These viruses can divide only in, that's multiply only in living cells. But he failed to demonstrate its particulate nature. Then he called this living that is in virus को इसने नाम दिया that's contagium vivum fluidium, means soluble living germs. Is it? In the early 20th century there was another English bacteriologist जिसका नाम था Twat. इसने डिस्कवर किया वायरस इन्फेक्ट बैक्टीरिया. जैसे हम आज पढ़ते हैं T2, T4, T5 दैट्स बैक्टीरियोफेज जो होते हैं इवन और ऑड, वो इसी ने ट्वाट ने एक्चुअली डिस्कवर किया था. दैट इज वायरस बैक्टीरिया पे भी अटैक कर सकते हैं. देन लेटर विद इन्वेंशन ऑफ इलेक्ट्रॉन माइक्रोस्कोप इन 1931 एंड द जर्मन इंजीनियर्स दैट्स रस्क एंड नल कैम फर्स्ट इमेज ऑफ वायरस. फर्स्ट इमेज ऑफ वायरस 1931 में आई जब क्या बनी? फर्स्ट इलेक्ट्रॉन माइक्रोस्कोप. In 1935 that's बायोकेमिस्ट एंड वायरोलॉजिस्ट जो था स्टैनली, इसने पहले एग्जाम किया दैट्स टबैको मोज़ेक वायरस एंड फाउंड इट्स मोस्टली मेड फ्रॉम प्रोटीन. This was the first scientist, that is Stanley, who first time demonstrated that this virus also is made from proteins. A shorter time later, this virus was spread into the protein and RNA parts also. Another at the time of that is 1931, another American pathologist जिसका नाम था that is Pasteur grew influenza and several other viruses in the fertilized chicken eggs. This is actually the discovery of virus. How virus discover actually you can study it easily on the book. That this Pasteur grew this influenza virus and other virus into the chicken egg. Now let's move towards the origin of viruses. That is how virus actually originate. The virus coexist with the life whenever it occurs. Means ये पहला consider किया जाता है जब when life arose here on earth, viruses also arose. The second point is viruses do not form fossils. So molecular techniques have been most useful means of hypothesizing how they arose. Means it is still in work. Three main theories actually come forward with the that is origin of viruses. Among them, the first theory here is that is this regressive theory. What's this regressive theory? This regressive theory states that virus may have once been small cells. Is it? That parasites the larger cells. मीन ये बताते हैं वायरस पहले small cells थी जो parasite करती थी on the large cells. ठीक है ना? What time genes do not require by their parasitism were lost. So genes जो need नहीं थे इस parasitism के लिए, so they lost. Is it? Bacteria, Rickettsia and Chlamydia are living cells that, like viruses, can reproduce only inside host cells. Means ये जो Rickettsia and Chlamydia और living cells, ये भी क्या हैं? Living cells. ये भी वायरस की तरह ग्रो करती हैं only inside the living cells. That is these two, Chlamydia and Rickettsia. These both reproduce only when there is living cell of any host. They lend credence to this theory, means they believe much more in this theory as their dependence on the parasitism likely to have caused the loss of genes that enable them to survive outside the host. मीन्स ये जो डिपेंडेंट थे ये पैरासाइट पे, इसी की वजह से लॉस्ट हो गए होंगे इनके जीन्स, और that's why they are enable them that this property enable them to survive outside the cell. The second theory here about the virus is that cellular origin theory. This theory states that some virus may have evolved from the fragments of DNA and RNA, that's or bits of DNA and RNA. These bits of DNA and RNA got escaped from the genes of larger organisms. जो larger organisms थे उनके genes से ये fragments of DNA RNA escape हो गए, and that is this escaped DNA could have come from these plasmids. As you know what's actually plasmid. So piece of DNA that can move between cells. So this plasmid is a piece of DNA. It can easily move between the cells and it can replicate sound, while the others may have evolved from the bacteria. This was the second theory, and another theory about the virus is that is this co-evolution theory. This states that virus may have evolved from the complex molecules of proteins and DNA at the same time as cells first appeared. Means this theory states that co-evolution means जब हमारा evolution हुआ उसी वक्त जब एनिमल्स प्लांट्स ह्यूमंस बने उसी वक्त इन वायरसेस का भी क्या हुआ, एवोल्यूशन हुआ. ठीक है ना? एंड would have depend on cellular life for many millions of years. This according to this co-evolution theory, as the name indicates, that's co-evolution. What's problems with these theories actually? The first problem with these theories that is the regressive hypothesis, which was the first theory, does not explain why even the smallest cellular parasite do not resemble virus in any way. करेक्ट इसका ये जो ड्रॉबैक है वो ये है दैट इज कोई भी ये जो पैरासाइट है वो रिजेंबल नहीं करता है वायरसेस के साथ. दिस इज द फर्स्ट ड्रॉबैक. The escape hypothesis does not explain the structure of virus particle. This is the second. And the third is that co-evolution or virus first hypothesis contravenes the definition virus in that they are dependent on the host. Simply जैसे हमें पता है viruses are mostly dependent on the hosts. Now let's study the that's characteristics of the viruses. ठीक है ना. हम देखते हैं वायरस के क्या कैरेक्टर होते हैं. फर्स्ट दे आर obligate intracellular. As I already told you that these viruses are intracellular means ये होस्ट की सेल में इन्वेड करते हैं and then start multiply. And so obligate intracellular parasite of bacteria, protozoa, fungi, plants, animals, algae, etc. Ultramicroscopic structure ranging from 20 nm to 450 nm in that is size. Not cellular in nature. They are not cellular in nature. Structure is compact and economical. Do not independently fulfill the characteristics of life. Means they are always dependent upon the host. Inactive macromolecule outside the host and active inside the host. Important point regarding the virus. Basic structure consists of protein shell, that's capsid, surrounding the nucleic acid core. ये हम देखेंगे अभी कैप्सिड क्या होता है. एंड न्यूक्लिक एसिड हमें पता है वायरस में होता है, दैट्स आरएनए और डीएनए हो सकता है. न्यूक्लिक एसिड कैन बी ईदर डीएनए और आरएनए बट नॉट बोथ. न्यूक्लिक एसिड दैट्स बोथ नहीं हो सकता. इट कैन ईदर बी डीएनए और आरएनए. It is some comparisons of the viruses with that's common cell. The first character is that structure. See here the structure of cell is that's virus cell and this common cell. That is this consists of DNA, RNA, and core of protein. That is sorry core of RNA, DNA, and this capsid. That's protection layer. But here is cell membrane, cytoplasm, eukaryotic also contain that's nucleus and organelle, etc. Reproduction only within the host cell, as I already told you. Independent cell division is there. Genetic code is that's DNA, RNA, but here is mostly genetic. That's DNA. Growth and development. Here is no. Here is absolutely yes. Obtain and use energy. Here is no, because they are dependent upon the host. But here is yes. Response to the environment. Pretty sure that it is no here. Why it's no? Because they are living inside the host. So इनका environment के साथ कोई भी ताल्लुक नहीं हो सकता. Yes, ये environment के response को stimulate कर सकती है. Change over time. Yes, this is same for the both. Another point is that is comparison is you can simply यहाँ पर स्क्रीन पे देख सकते हो. This is surface protein, that's this layer, and this is protein shell, that's this capsid, and this is genetic material, that's this. But simply you can see that cell, normal cell, इसमें न्यूक्लियस है, साइटोप्लाज्म है, राइबोसोम्स हैं, माइटोकॉन्ड्रिया हैं, एटसेट्रा हैं. ठीक है ना. This is the comparison between viruses and other cells. Now let's talk about the virus size. What? So size में हम पहले देखते हैं that's viron size ranges 10 to 400 nm, as I already told you. All the virons contain nucleocapsid, which is composed of nucleic acid and DNA. That's nucleic acid, DNA भी हो सकता है, RNA भी, and protein code, that's capsid. First मैं आपको बताऊं पहले viron क्या होता है. It's a simple virus particle. ठीक है ना. Simple virus particle often designed virus. Sorry, viron. Is it? Now, some virus consists of only a nucleocapsid. Others have additional components. वो हम देखते हैं additional components किन वायरस में होते हैं. So here is some size of some viruses, that is bacteriophage अगर हो 225 nm, that's rabivirus 170 into 70 nm, that is adenovirus, that's 90, rhinovirus, that's 30, and here is bacteriophage, that's 800 into 10 nm. Here is also bacteriophage F2ms2, that's 24. Here is poliovirus, that's 30, prion, that's 200 into 20, and here is vaccinia virus, it is 300 to 200 into 100 nm. E. coli is here, that is bacterium, its size is larger than virus. So this is also one comparison between viruses and the bacteria. Now let's see what's nucleocapsid in viruses. Nucleocapsid is that's the capsid and the nucleic acid together called nucleocapsid. Nucleo as is that's nucleic acid and capsid means प्रोटीन कोट, वो हम देखते हैं अभी. वायरन जैसे मैंने आपको बताया था fully formed virus that's able to establish an infection in the host is known as viron, and this is capsid actually. See here, this is capsomer and this is capsid. So what's different between capsid and capsomer? Capsomer is single unit, and when these all units are together, these are known as capsid. Is it? And this is nucleic acid. That's this. It can either be RNA or DNA. So what's the function of this capsid is it provides protection to the nucleic acid of virus. ठीक है ना? ये nucleic acid virus का जो होता है इसको protection करता है. That's called capsid. Is it? Now in this picture you can also see here is viral components is three, that's nucleic acid, capsid and envelope. What's envelope? Let me tell you. मैंने आपको पहले भी ये बताया था ये फैट का बना होता है mostly. So capsid composed of many capsomers, that's this is capsid, and this is nucleic acid DNA core, and this DNA is protected by this capsid. Is it? Now this is envelope, that's this layer. It's only present in some viruses, but some viruses lack this envelope. Is it? And these are spikes. What is the function of these spikes? Help in attachment to the host. These spikes help virus to get attached with the host. What's genome actually present in viruses? जो मैं बता रहा था आपको RNA, DNA, वही हम अब देखते हैं genome. The sum total of genetic information carried by an organism is genome. First, the number of viral genes compared with the cell is quite similar. Is it? इनके पास वही genes होते हैं जो इसे help करते हैं to multiply inside host. Is it? They only have the genes necessary to invade the host cells and redirect their activity. जो मैंने अभी आपको बताया. This genome is actually two types in bacteria. That is, first is that is DNA viruses. DNA can be either single stranded or double stranded. What's single stranded and double stranded? Look at this picture here. That is this. देखो यहाँ पर DNA viruses हैं. DNA viruses can be enveloped or non-enveloped. Enveloped viruses is mostly double-stranded. That is double-stranded. These contain mostly double-stranded genome. Its example is pox virus and herpes virus. But these non-enveloped can be either double-stranded or single-stranded. Double-stranded is that is adenovirus. Here is this papovirus and this is parovirus. This parovirus is quite single-stranded genome. Is it? And here is another type of nucleic acid found in that is viruses, that is RNA. Mostly single-stranded. पॉजिटिव, इसमें जो आरएनए वायरस होते हैं इनमें दो स्ट्रैंड्स होते हैं, दैट्स पॉजिटिव सेंस एंड नेगेटिव सेंस. दिस इज इंपॉर्टेंट टू रीड हियर. दैट्स पॉजिटिव सेंस क्या होती हैं? जीनोम दैट आर रेडी फॉर इमीडिएट ट्रांसलेशन इनटू प्रोटीन. मींस ये इमीडिएट प्रोटीन की ट्रांसलेशन करते हैं. दैट इज होस्ट की सेल में जो इनको जो भी जरूरत चाहिए, दैट इज राइबोसोम एटसेट्रा, कोई भी उसे हाईजेक कर लेते हैं, फिर ये इमीडिएट प्रोटीन सिंथेसिस करना स्टार्ट कर देते हैं. बट दिस नेगेटिव सेंस आरएनए इज दैट इज genome have to be converted into proper form to be made into protein. Means this is not quite functionally at the time of infection. So इससे पहले कुछ modifications करनी है खुद में, then it can synthesize its genome, sorry, proteins. Now here is RNA viruses, like that is DNA viruses. They can be enveloped and non-enveloped. Enveloped can be either single-stranded or single-stranded genome encoded with reverse transcriptase. Is it? The first single-stranded genome is found in segmented genome, and this single-strand genome can be segmented or non-segmented. Segmented is found in these types of viruses, and non-segmented is found in these type of viruses. Here that is adenovirus, that is orthomysovirus, etc. Here is that is parvovirus or coronavirus or paramozovirus, etc. This is a non-segmented genome in these viruses. On the other hand, enveloped is also that is single-stranded genome encoded reverse transcriptase, that is retroviruses में जो हमें पाया जाता है मोस्टली, दैट इज इनमें रिवर्स ट्रांसक्रिप्शन होती है. दिस इज एनवलप्ड टाइप ऑफ वायरस. नॉन एनवलप्ड में कौन कौन सा होता है? दैट इज सिंगल जीनोम, सिंगल स्ट्रैंड जीनोम एंड डबल स्ट्रैंडेड जीनोम. सिंगल स्ट्रैंडेड में कौन कौन सा होता है? दैट्स पाइकोरना वायरस एंड कैल्सी वायरस. यहां पर इसकी एग्जांपल है रियो वायरसेस. Here is picture, that is you can simply see here. This is enveloped virus. This is also enveloped. This is enveloped. ये envelope है. This is envelope here, and these are naked viruses. Means they don't contain any envelope. In this picture, you can also see envelope here. This is capsid. This is nucleic acid here, and this is spikes, and this is envelope. Is it? This is envelope. Here is also capsid, nucleic acid, but it's naked. इसमें envelope present नहीं है. Tools for studying structure of virus. The first tool here is that is electron microscope. We need excellent microscope with high resolution. Is it X-ray diffraction or cryo electron microscope is also helpful to study the virus. Now let's study first what's actually this capsid. Capsid is constructed from identical subunits, that's capsomers. मैंने पहले भी आपको बताया. It's mostly made up of protein molecules. ठीक है ना? It's of two types, that is helical. Helical can be rod-shaped capsomers assembled into helical nucleocapsid. ठीक है ना? Nucleo means nucleic acid. Capsid means that's these multiple units of capsomers. This is capsid. In this picture, you can easily see it here. These are the capsomers. 1, 2, 3, 4, 5. These different or multiple are capsomers, and this is nucleic acid here. And together we call them capsid, helping protection of nucleic acid. So this is capsid. You can also see in this picture here, that is nucleic acid, capsomers, and collectively we can call them capsid. What's actually function of this capsid? It protects nucleic acid. The second is help introduce the viral DNA or RNA into the suitable host. It's the second function of this, that is the capsids or capsomers. Stimulate the immune system to produce antibodies that can protect the host cell against the future infections. This is the third function of virus capsid. And here you can also see this is the nucleic acid and these are the capsomers and this is capsid. This is actually Ebola virus. And this is whole about the general introduction of viruses. Or you can also see here in the bacteriophages that this is capsid here and this is DNA genetic material present here. Is it this is sheet? We will study bacteriophage हम अलग से पढ़ेंगे. But here I have to tell you some points about the viruses, that is जो plants में viruses अटैक करते हैं, these contain the genetic material only RNA. ठीक है ना, that single strand RNA उन viruses में होता है जो plants में virus करते हैं. The second point is that animal viruses जो होते हैं, एनिमल वायरसेस में इधर इनमें डीएनए होगा जेनेटिक मटेरियल, आरएनए होगा, सिंगल स्ट्रैंड आरएनए हो सकता है एंड डबल स्ट्रैंड आरएनए भी हो सकता है इन केस ऑफ एनिमल्स. अनदर थर्ड पॉइंट में आपको ये बताऊं, दैट इज बैक्टीरियल वायरस जो होते हैं, दैट्स बैक्टीरियल वायरस और बैक्टीरियोफाज वी कैन सिंपली कॉल देम. इनमें क्या होता है, मोस्टली डबल स्ट्रैंडेड डीएनए होता है. डबल स्ट्रैंडेड डीएनए, दे यूजुअली कंटेन डबल स्ट्रैंडेड डीएनए, but can also have single stranded RNA and single stranded DNA. ठीक है ना, single stranded DNA also or RNA can also be present in viruses. Fourth point here is that insect वायरस जो होते हैं, कौन, इंसेक्ट वायरस में क्या होता है, these contain RNA, इनके पास RNA जेनेटिक मटेरियल होता है एंड बट फ्यू के पास क्या हो सकता है, DNA भी जेनेटिक मटेरियल हो सकता है. दिस इज also important regarding the virus, and this was all about the introduction of viruses.